Chapter 4 covers nucleic acid extraction methods. The chapter covers isolation from DNA and also touches on RNA isolation. The objectives covered include comparing and contrasting organic, inorganic, and solid phase approaches for DNA isolation, comparing and contrasting organic and solid phase approaches for isolating total RNA, distinguishing between the isolation of total RNA with that of messenger RNA, describing the gel-based, spectrophotometric, and fluorometric methods used to determine the quantity and quality of DNA and RNA preparations, and calculating the concentration and yield of DNA and RNA from a given nucleic acid preparation. Nucleic acid is routinely isolated from human, fungal, bacterial, and viral sources in the clinical laboratory. See Table 4.1 in your textbook that shows a summary of the yield of DNA from different specimen sources. Pretreatment may be necessary to make cells available from which nucleic acid will be extracted depending on the type of specimen. For example, white blood cells in blood or bone marrow specimens are removed from red blood cell and other blood components by either centrifugation or lysis. Fresh or frozen tissue sample must be dissociated before DNA isolation procedures. Grinding, homogenizing, or mincing tissue are methods to disrupt the whole tissue. Fixed and embedded tissues that are processed in histology, may be deparaffinized by soaking in xylene. The DNA quality depends on how the tissue is handled prior to fixation. Some bacteria and fungi have tough cell walls that need to be broken down to release the nucleic acids. Enzyme products can be used along with detergent treatments and similar products to access the nucleic acids in the microorganisms. Different commercially available agents may also need to be used for isolation of DNA to be used in amplification procedures such as PCR. Specimens such as dried blood, saliva, bone, teeth, amniotic fluid, hair, buccal cells, CSF, fixed tissues, feces, and soil can be used for DNA isolation and amplification procedures. As mentioned on the previous slides, DNA can also be extracted from fixed specimens. These type of specimens are usually samples from anatomic pathology or histology preparations. Depending on the fixative used, DNA may be broken or it may be uncross-linked to varying extents. The best type of fixatives are acetone or 10% buffered neutral formalin. Zambonis, Clark's, paraformaldehyde, formalin alcohol acetic acid, and metharkin are not as good fixatives for molecular testing. The least desirable fixative are carnoys, Zenkors, bouillons, or B5. After the release of DNA from the cell, further purification requires removal of contaminating proteins, lipids, carbohydrates, and cell debris. There's different DNA extraction methods. Organic extraction methods use a combination of high salt, low pH, and an organic mixture of phenol and chloroform. Inorganic methods do not require phenol extraction and were developed due to safety concerns with the caustic reagents such as phenol that make it a less desirable method. Inorganic methods are also known as salting out and use low pH and high salt to selectively precipitate proteins, leaving the DNA in the solution. The solid phase extraction method involves a more rapid and comparably effective technique using solid matrices to bind and wash DNA. Examples of solid phase include silica-based products such as columns or beads. Before performing the extraction, the specimen needs to be prepared appropriately. Preparing blood or bone marrow samples for DNA isolation involves either centrifugation or lysis. With centrifugation, the sample is mixed with isotonic saline and then overlaid with phycol. Phycol is a highly branched sucrose polymer that does not penetrate biological membranes. Upon centrifugation, the mononuclear white blood cells, such as lymphocytes and monocytes, which are the desired cells for isolation of nucleic acid, settle into a layer in the Ficol gradient. They're below the less dense plasma components and above the polymorphonuclear cells, such as neutrophils, eosinophils, basophils, and then also the red blood cells. The layer with the mononuclear cells is then removed and washed by rounds of resuspension and centrifugation in saline before proceeding with the nucleic acid isolation procedure. Lysis is done using differential osmolysis, which takes advantage of the osmotic fragility of red blood cells and white blood cells. Incubation of whole blood or bone marrow in hypotonic buffer or water will result in lysis of red blood cells before white blood cells. The white blood cells are then pelleted by centrifugation, leaving the empty red blood cell membranes and hemoglobin, respectively, in suspension and solution. With tissues, the samples can be frozen, then crushed, or fresh tissue can be grinded, homogenized, or minced in order to prepare for extraction. Enzymatic digestion using enzymes such as proteinase K may also be used to prepare these types of samples. Finally, plants or fungi may be broken down mechanically by homogenized or vigorously mixed with glass beads to prepare specimens for DNA isolation. This figure is a general schematic of DNA isolation by organic methods. Once the sample is prepared, DNA isolation is performed in several steps. First, lysis, in which the cells and nucleus from the prepared specimen are broken open to release the DNA using sodium hydroxide and sodium dodecyl sulfate, also known as SDS, a detergent. This all occurs in a microtain or a test tube. Step 2, acidification with acetic acid and salt serves to lyse red blood cells if present in the specimen and also to neutralize the pH, allowing DNA strands to renature. Step 3, the extraction, is when a biphasic emulsion forms when phenol and chloroform are added to the suspension of lysed cells. At the proper pH, centrifugation will settle the hydrophobic phase on the bottom. This includes all dissolved lipids and other hydrophobic components, and DNA will dissolve in the upper aqueous phase. The last step, precipitation, is when DNA in the upper aqueous phase is then precipitated mixing with ethanol or isopropanol and salt. The DNA precipitate is collected by centrifugation. Excess salt is removed by rinsing the pelleted nucleic acid, centrifuging, and then discarding the supernatant and reserving the pellet. This schematic shows inorganic DNA isolation. Remember, I mentioned before there are safety concerns with caustic reagents such as phenol, so inorganic DNA isolation methods were developed. Initially, inorganic methods did not provide the efficient recovery of clean DNA achieved with phenol extraction. However, newer methods have proven to produce high-quality DNA preparations in good yields. Low pH and high salt conditions selectively precipitate proteins, leaving the DNA in solution. The DNA can then be precipitated as described in the organic method using isopropanol pelleted and resuspended in TE buffer or in water. Preparation of samples for isolation of DNA on solid phase media starts with cell lysis and release of nucleic acids, similar to organic and inorganic procedures. Specific buffers are used to lyse bacterial, fungal, or animal cells. Buffer systems designed for specific applications are commercially available. For solid phase separation, the cell lysate is applied to a column in high salt buffer, and the DNA in solution absorbs to the solid matrix. After the immobilized DNA is washed with buffer, the DNA is eluted in a specific volume of water, TE, or other low salt buffers. Of note, TE is the tris EDTA buffer. The washing solutions and the eluent can be drawn through the column by gravity, vacuum, or centrifugal force. DNA absorbed to the magnetic beads is washed by suspension of the beads in the buffer and collection of the beads using a magnet applied to the outside of the tube while the buffer is aspirated or poured off. Solid phase DNA isolation media includes silica, spin columns, and beads. Nucleic acid binding to silica beads is the basis for many automated extraction systems, such as the Roche MagnaPure and Qui-Gon BioRobot. These use magnetized glass beads or membranes to bind DNA. These systems are finding increased use in clinical laboratories for automated isolation of DNA from blood, tissue, bone marrow, plasma, and other body fluids. Isolation of DNA from limited amounts of starting material, such as dried blood or bone, and isolation of DNA from challenging samples, such as fixed, paraffin-embedded tissues, can yield sufficiently useful DNA for amplification procedures. The analysis of paraffin samples is frequently performed in the clinical laboratory. Fixed tissue is more conveniently accessed in the laboratory and may sometimes be the only source of patient material. Thin sections are usually used for analysis, although sectioning is not necessary with very small samples, such as needle biopsies. Paraffin-embedded specimens must be dewaxed with xylene or other agents and then rehydrated before nucleic acid isolation. Before lysis, cells may be washed by suspension and centrifugation in saline or other isotonic buffer. Reagents used for cell lysis depend on the subsequent use of the DNA. For simple screens, cells can be lysed in detergents such as SDS or Triton. For use in PCR amplification, cells may be lysed in a mixture of tris buffer and proteinase K. The proteinase K will digest proteins in the sample, lysing the cells and inactivating other enzymes. The released DNA can be used directly in the amplification reaction. With the advent of PCR, new and faster methods for DNA isolation have also been developed. The minimal sample requirements of amplification procedures allow for the use of material previously not able to be used for analysis. Rapid lysis methods provide sufficiently clean DNA that can be used for amplification. DNA purification can be performed with Chelex resin for PCR testing. Chelex is a cation chelating resin that can be used for simple extraction of DNA. A suspension of 10% Chelex resin beads is mixed with the specimen, and the cells are lysed by boiling. After centrifugation of the suspension, DNA in the supernatant is cooled and may be further extracted with chloroform before use in amplification procedures. This method is most commonly used in forensic applications, but may also be useful for purification of DNA from clinical samples and fixed paraffin-embedded specimens. This schematic is just an example showing DNA extraction from fixed tissue for PCR. Here we have sections from a histological preparation that are then macro or micro dissected and can be used for digestion with proteinase K or tris buffer. After the sample is processed, then it can be used for PCR testing. This procedure is an example of DNA extraction and storage using Whatman FTA paper for PCR. The Whatman FTA card is a filter paper product manufactured by GE Healthcare. It's a paper matrix laced with proprietary mixtures of chemicals that lyse cells and stabilize nucleic acids on contact for long-term storage at room temperature. So in the procedure here you can see the first step is the spot test, applying a sample and allowing it to completely dry and then store for future processing. Next, punching out a piece of the filter paper and removing a disc from the center of the dried specimen for testing. An example of testing that uses this type of methodology is newborn screening using blood spot to test for various disorders that can be hereditary due to infection or caused by maternal medical problems. Mitochondrial DNA can also be isolated for testing. There are two approaches to the isolation of mitochondrial DNA from eukaryotic cells. One method is to first isolate the mitochondria by centrifugation. After cell preparations are homogenized by grinding on ice, the homogenate is centrifuged at low speed to pellet intact cells, nuclei, and cell debris. The mitochondria can be pelleted from the supernatant in a second high-speed centrifugation. The mitochondria can be lysed with detergents and the lysate treated with proteinase to remove protein contaminants. Mitochondrial DNA can be precipitated with cold ethanol and resuspended in water or appropriate buffers for analysis. The second approach to mitochondrial DNA preparation is to isolate total DNA. The procedure is similar to that described in the centrifugation method. The preparation will then contain mitochondrial DNA that can be analyzed within the total DNA background by hybridization or by PCR. After DNA has been isolated, the next thing to discuss are the methods to assess the DNA. Gel electrophoresis is a technique used to separate DNA fragments according to their size. DNA samples are loaded into wells, which are little indentations at one end of the gel, and an electric current is applied to pull them through the gel. DNA fragments are negatively charged, so they move towards the positive electrode. Because all DNA fragments have the same amount of charge per mass, small fragments move through the gel faster than large ones. When a gel is stained with DNA binding dye, such as ethidium bromide or Sibir green 2, the DNA fragments can be seen as bands, each representing a group of same-sized DNA fragments. Genomic DNA should appear as in the image. It runs as tight smears close to the loading wells. DNA yield can also be assessed using spectrophotometry to measure the absorbance. One optical density unit or absorbance unit at 260 nanometers is equivalent to 50 micrograms per milliliter of double-stranded DNA. To calculate the yield of DNA after preparation, multiply the DNA concentration times the total sample volume. To calculate the purity of the DNA sample, measure absorbance from 230 nanometers to 320 nanometers to detect other possible contaminants. The most common purity calculation is the ratio of the absorbance at 260 nanometers divided by the reading at 280 nanometers. Good quality DNA will have an absorbance ratio of 1.6 to 2.0. A reading of less than 1.6 does not render the DNA unsuitable for any application, but lower ratios indicate more contaminants are present in the sample. This next section of Chapter 4 focuses on nucleic acid extraction methods for RNA. Working with RNA in the lab requires strict precautions to avoid sample RNA degradation. RNA is especially labeled due to the presence of RNases that are found in both prokaryotes and eukaryotes and in practically every cell type. RNases are enzymes that are small proteins that can renature even after autoclaving and regain activity. Unlike DNases, RNases need to be eliminated or inactivated prior to isolation of RNA. It's necessary to designate a separate RNase-free area of the lab for storage of materials and specimen handling intended for RNA analysis. Disposables, reagents, etc. must all be designated DNase-free and RNase-free by suppliers. RNA tests, especially those that involve gene expression, such as array analysis or quantitative reverse transcriptase PCR, are more accurate when the RNA is stabilized from further metabolism or degradation after collection. We'll talk more about acceptable tube types in Chapter 16, but EDTA, the lavender top tube, then ACD, the yellow top tube, are the recommended tube types for molecular testing, as heparin, the green top tubes, have been shown to inhibit enzymes used in molecular analysis. So the types of specimens that can be collected include bone marrow and peripheral blood, and then tissue samples. Specimen preparation is similar to the procedures discussed in the DNA extraction methods section, but differs in some respects, remembering that RNases need to be inactivated. Red blood cells in blood and bone marrow are lysed by osmosis or separated from white blood cells by centrifugation. Tissue can be frozen, then crushed, or fresh tissue can be grinded, homogenized, or minced, but immersed in buffer that will inactivate intracellular RNases. This is especially true for tissues such as those from the pancreas that contain large amounts of innate RNases. Bacteria, fungi, and plants can be prepared using enzymatic digestion or may be broken down mechanically by homogenized or vigorously mixed with glass beads to prepare the specimens. RNA isolation methods include organic, which uses organic chemicals such as phenol and chloroform, and solid phase, that utilizes solid support beads or columns in which the DNA is immobilized. In the organic RNA isolation procedure, the first step is cell lysis. This is performed in detergent or in phenol in high salt or an RNase inhibitor. The RNase inhibitor used is guanidine isothionate, also known as GITC. This is a strong denaturant of RNases that can be used instead of high salt buffers. The next step is extraction. Once the cells are lysed, proteins can be extracted organically with phenyl chloroform isomal alcohol solution. The chloroform enhances the extraction of nucleic acids by denaturing proteins and promoting phase separation. Isomal alcohol prevents foaming. For RNA, the organic phase must be acidic, with a pH of between 4 to 5, so acidity is adjusted using pH-appropriate buffer. After phase separation, the upper aqueous layer containing the RNA is removed to a clean tube. Precipitation of RNA occurs by the addition of ethanol, in which pellet formation occurs. The RNA precipitate is then washed and resuspended in RNase-free buffer or water. Solid phase separation of RNA begins similarly to the steps described in the organic extraction method, but instead the cell lysate is applied to a column and the RNA in solution absorbs to a solid matrix. Washing solutions and the eluent are drawn through the column by gravity, vacuum, or centrifugation. The eluated RNA is usually sufficient concentration and purity for direct use in most applications. Automated systems use magnetic beads or particles for RNA isolation as well. The quality of RNA from fixed paraffin-embedded tissue depends on the fixation process and handling of the specimen before fixation. Commercial reagents are designed to provide lysis and incubation conditions that reverse formaldehyde modification of RNA and efficiently release RNA from tissue sections while avoiding further RNA degradation. This slide revisits the types of RNA that we saw in Chapter 2. These are different types of RNA that can be extracted and analyzed for testing. Messenger RNA, mRNA, is the form of RNA in which genetic information transcribed from DNA as a sequence of bases is transferred to a ribosome. Ribosomal RNA is a component found in ribosomes essential for protein synthesis in all living organisms. Transfer RNA is RNA consisting of folded molecules which transport amino acids from the cytoplasm of a cell to a ribosome. Heteronuclear RNA is the RNA before splicing occurs. Small nuclear RNA is involved in splicing or other gene processing reactions. Double-stranded RNA is involved in gene silencing. MicroRNA is a regulatory RNA. Small or short interfering RNAs are functional intermediates, and many short RNAs, such as snRNA or tncRNA, influence various cellular processes. Often, the RNA needed for analysis is messenger RNA, which accounts for up to only 2.5 to 5% of the total RNA yield. To enrich the yield of messenger RNA, protocols employing single-stranded oligomers of thymine or uracil, as seen in this figure, are immobilized on a matrix resin column or beads are used. The poly-T or poly-U oligomers will bind the poly-A tail found exclusively on messenger RNA. After washing away residual RNA, poly-A RNA is eluated by washing the column column to break the hydrogen bonds between the messenger RNA and the column. The last slide discusses the methods to assess the RNA once it's been extracted. High-quality total RNA appears as two distinct rRNA bands when assessing by gel electrophoresis. This will be discussed in more detail in Chapter 5. RNA yield can also be assessed using spectrophotometry similar to DNA to measure the absorbance, the optical density. One optical density unit at 260 nanometers is equivalent to 40 micrograms per milliliter of RNA. To calculate the yield after preparation, multiply the concentration times the total sample volume. To calculate the purity of the sample, measure the absorbance from 230 nanometers to 320 to detect other possible contaminants. The most common purity calculation is the ratio of the absorbance at 260 nanometers divided by the reading at 280. Good quality RNA will have an absorbance ratio of greater than 1.6. One other method to assess RNA is fluorometry, or fluorescence spectroscopy. It measures fluorescence related to concentration and can be used for DNA or RNA. Cybergreen 2 RNA gel stain intensity of a fluorescence is 20 to 26% lower with polyadenylated RNA than with total RNA. Of note, this dye is not specific to RNA and will bind and fluoresce with double-stranded DNA as well. Contaminating DNA must be removed to get an accurate RNA concentration when using this method to assess it. So, in summary, DNA can be extracted using organic, inorganic, and solid phase methods. It can also be extracted by more rapid methods or methods designed for challenging specimens, such as the dried blood samples using the Whatman filter paper. RNA extraction methods include organic and solid phase methods. Messenger RNA can be specifically extracted using immobilized poly-T or poly-U. And finally, DNA and RNA concentration, yield, and purity can be assessed using either gel electrophoresis analysis, spectrophotometry, or fluorometry.